Passive and active transport has to do with concentration gradients, and bulk transport involves endo and exocytosis. So first of all, solute concentration. Solute is just a substance suspended in water, and when we talk about concentration, we're talking about the amount. So high concentration has a lot of solute, low concentration has a little solute. When we talk about gradients, a lot of times I like to think about steep grade ahead, like a sign you might see out west. And we have this steep hill. So that gradient, we have a concentration gradient where the high amount is at the top and the low amount is at the bottom. If we're going to talk about passive transport, we're going to talk about substances traveling from high to low concentration. We might say that's down or with the concentration gradient. And they are naturally going to flow that way, and no ATP energy is needed for passive transport. When we reach a balanced amount, there is no concentration gradient, but we would still have some molecular movement back and forth. It's not like everything just freezes. If we go back to that concentration gradient, and this time we want to go up or against the concentration gradient, we are going to call that active transport. And if we are heading against that concentration gradient, it would be difficult to move those molecules up the hill, so ATP energy is needed. Another way I like to think about this is substances and how they would naturally go and whether they would need energy to do so. So these molecules, if we drop some dye into a fish tank, that dye would naturally diffuse and spread evenly and be balanced throughout the fish tank. In that case, no ATP energy would be needed on the part of the fish to allow the molecules to disperse. That would be passive. The goldfish would be passive. Now, if we imagine it in reverse, and if this were actually possible, imagine the energy it would take for these goldfish to try and move all of the dye into a individual spot. That would take, the goldfish would be really have to be active if it were actually possible to gather up each individual molecule of dye and keep them in one place. ATP energy would be needed in order to make things more crowded because they would naturally want to just spread out. So when we look at these different types of molecules, we're going to have passive and active transport with ions and a lot of molecules. But when we start talking about bulk transport, we're talking about large amounts or large molecules. So for bulk transport, ATP energy is needed, and we're going to involve vesicles. So in the case of exocytosis, a lot of times when we think about this, we think about vesicles created from the rough endoplasmic reticulum, little packages of proteins that will bud off of the rough ER, go through the Golgi apparatus, and ultimately exit the cell. So this plasma membrane is made from the membrane of the rough ER. And what it's going to do is those membranes are going to merge with the outer cell membrane, and this vesicle membrane ultimately becomes part of the outer cell membrane, and the contents are then exited from the cell. In the case of endocytosis, substances are brought into vesicles. Again, this is still bulk transport. We can have phagocytosis, which is cell eating, and part of that outer plasma membrane becomes part of the vesicle membrane as it pinches it off as it comes in. We can have also pinocytosis, which is like cell drinking, and receptor-mediated endocytosis, where the solute actually binds to receptors and then forms a coated vesicle. So exo is out and endo is in. Passive and active transport deal with concentration gradients. Exo is out, endo is in, both bulk transports that requires energy.